Thank you so much, Pat, for that introduction. I probably will never talk about my research as well as Pat just did, but I am an aquatic and evolutionary biologist. I'm based out of Calgary, which you might be wondering how someone can be landlocked and still be interested in all of these marine organisms. And the answer to that is I mostly go and do a lot of field work on the West Coast on Vancouver Island, but I've been expanding my research program since settling on the prairies to include a lot more aquatic organisms that are in freshwater systems. And then I've also started branching out into plants and algae and really anything that's kind of attracted my interest. And a big part of that is what my students have been interested in as well. So Pat just gave a huge overview about some of the projects that I've been doing. But really, it kind of comes down to this idea that in my job, I'm really passionate about quantitative biology. All of the stuff that I do in field research and that we do in field ecology is messy data because animals and plants do weird things. And it's really hard to predict what they're going to do and what those responses are going to be. And one of the ways that I've found out of necessity that's helped me do those things is a quantitative approach to the analyses with all that data that I bring back. I'm also really big into science communication and being able to clearly articulate the science that we do. I love talking about it with everybody, whether they have a science background or not, and being able to do that, I think, is a really key skill in the ways that we kind of go through our careers. You can come in, it's okay. And I also love talking about different science careers and pathways. I think that's a really big part of my work is just letting everybody know what is available during your undergrad, but then also afterwards and where you're going. And so a lot of the work that I'm doing now as I transition into kind of more biomes that I haven't had as much experience in takes place in these really beautiful prairie grasslands, a little bit more wetland work and freshwater systems, because these areas are only 15 minutes from my campus in Calgary. So I actually have access to a ton of new environments. And it's really helped me expand the questions that I'm asking and the places in which I can conduct that work. The work I'm going to talk about today usually looks a lot like this with a whole bunch of undergraduate students. In particular, I do a lot of this work at the Banfield Marine Sciences Center through their summer programs with undergraduate student researchers and also in their fall programs, which is a cohort-based fall program that you can go out to Banfield and take courses there for 4 months. And the work that we do out at Banfield is all driven by the students and what they're interested in. And so it keeps my research program as exciting as possible because right now I'm working with students on prawns and on crabs and on algae, and it's all just based off of the things that they brought questions to me about. And that's where our research went. The only way, though, that we can do this type of research is with really intentional and reciprocal, respectful relationship building with the Indigenous nations that live in the area. And the Hawaiian First Nations have been the traditional stewards of the land that the Banfield Marine Sciences Center sits on now. All of the work that we do is in their halhuthi, or those traditional territories and waters. And my research has been really kind of driven by their 3 sacred principles that this modern treaty nation governs themselves by as well. And those principles are Isak, or the greater respect for the world that we live in, the Hishuk Masawak, everything is one. A lot of times in ecology, we think about like the plants and the animals and how they're interacting, and we always take ourselves out of the equation. We're just really impactful, odd behaving animals as humans. And so bringing ourselves back into that ecosystem and understanding the impacts that we can have on our ecosystems, thinking about how everything is one and interconnected, is a really important part of my work. And also uathak, or taking care of the stewardship of the land and understanding how we can preserve the functioning of the ecosystems that we work in and that we live in. And so my program at Mount Royal University is entirely driven by undergraduate researchers, and I try to bring as many of those experiences to as many undergrads as I possibly can. And so in terms of why I think these things matter, and hopefully if I can convince you why research experiences might matter while you're in your undergraduate degree, is that it is more than just a field trip, right? We love getting out there. It's beautiful in these places that we can go and do this work in, but it's more than just going into the field for a cool hike. Being able to engage in research projects with where you're at in your careers really does develop those critical thinking skills that employers are looking for, because you're able to not just think about something that you've observed in the field, but then you can start asking questions about it and be critical. Why is that thing doing it the way that it's doing it? How do we understand how that might be impacted by the environment that we find it in? What happens if we put it in a different environment? Those types of skills, that problem solving, that critical thought about the observations that you're having, those are all integral to how we start our research projects. There's also an ownership of your learning. As much as possible, I will try to provide the guidance for the project, the question, the topic, maybe a method or two that's useful. And then you go out and do exactly what you want to do. I will let you know if it's like, ooh, 3 hectares a day, that's going to be too much. Well, there's some guidance there for sure. It's not just trial by fire. But the questions that you're asking and the way in which you're approaching it, I want to be and should be as much your own as possible. So, at least for me, the motivation for when I'm doing my science, when it's a question I'm really stoked about because it's something that I was like, that thing's weird, I want to figure out what it's doing. When I have that ownership over my own learning, I find that I'm much more motivated to do things. There's also professional networking involved. A lot of the reasons that we can go out and access these really beautiful areas and ecosystems is because we have bases, like a field station, that are run by professional scientists and administrators that help us do the science that we want to do. So I'm going to talk about the work that we did at the Banfield Marine Science Center last summer with a group of undergraduate researchers from McMaster, of whom I see here today. And then I'm also going to talk about one of the field stations that we're going to go to this summer, and the support that you get, the other scientists that you meet, and the work that you're able to do because of the relationships that you build. It's all part of that ability to network and start to know people in your field. It's also a big element of doing undergraduate research is the preparation that you get for your career. These are skills that we might not implicitly be teaching you in classes, but these are things that are going to be really, really helpful for when you're on the job market and when you're able to define and acknowledge those transferable skills, like being able to critically evaluate papers, to pull information from multiple different sources, designing an experiment, and when it doesn't work, knowing where to go next with it. All of that kind of troubleshooting stuff is going to be so key for wherever you end up going next in your career, whether it's academia or industry or government. And then one of the things I can tell you how impactful these experiences are, and for myself when I was an undergraduate, for the students that I have going through, but then we noticed that there was such an interesting kind of, everybody had the same sort of experience, that these undergraduate research experiences were really helping students grow professionally and personally as they went through them. And so people have done science and studied that as well. And students have really reported that some of the largest changes in their undergraduate careers has been through research experiences in terms of their growth and their ability to feel really self-reliant and their ability to then go forward and think, I have the skills, I have the ability to tackle these new challenges. And we get to do that more or less all outside when we're doing field work. And so it is a field trip, but there's also a lot of really great elements from a personal and professional development standpoint that undergraduate research can offer you. And so you might be wondering why I am standing here from Calgary, Alberta, but I'm talking about working at the Banfield Marine Sciences Center, and we're in Ontario at McMaster University right now. And how did this all come together? So if you've been around McMaster for the last little while, you may have met Abir Siddiqui, a science librarian. And Abir and I actually met when we were both working at the Banfield Marine Sciences Center over a decade ago now. And when Abir and I were both working there, we've continuously talked about going back and working together. And then Abir got in touch with me last year and said, I had this amazing colleague at McMaster named Sunita, who has done a lot of work at Banfield in the past as well and is really interested in getting students out to the station. Like, how can we do this? And so Sunita and I got connected through Abir, and we started talking about what it might look like to bring students out to the Banfield Marine Sciences Center, which is where this wonderful group of students came into play last year. Sunita was able to bring together these students who had applied and brought them all out to the Banfield Marine Sciences Center and met me. I was already been out there for about 3 or 4 weeks at that point. And I got to meet this incredible new group of students that was coming out to do research with me. So I want to talk a little bit about the research that they were doing because I think it was really cool. And I analyzed a lot of the data from the experiments that they did in order to show you what they were doing. So hopefully for the ones who did all this research, I hope you're excited to see what you found as well. We were working on the European green crab, which in Europe, as you may have guessed from the name, it's a native species. Its population is under control based on the evolutionary history that it's had in that area where it's been able to kind of find its niche. It has predators that control its population. It has food sources, but it never really gets too big of a population or too small of a population. It's kind of held in check. It was introduced to Canada along the East Coast, first into the US, and then it migrated up into Canadian waters. And there have been multiple introductions along the East Coast of Canada, and then an eventual introduction to the West Coast of Canada as well. And that happened in kind of 3 major introductions. We had a population that came from the eastern seaboard and migrated upwards into Canada, but it was not really a cold-tolerant crab population at that point, so it didn't get too much of a foothold in most of the Maritimes. Then we had another population come over from Northern Europe and establish in northern Nova Scotia, which meant that we had a cold-tolerant crab population. And then it hybridized with a not tolerant population. And then they made this like 3rd hybrid super crab population that was able to handle both warm temperatures and cold temperatures. And then we got another introduction to the West Coast, but much, much later. So we had these introductions coming in around the 1950s. We didn't get European green crabs on the West Coast of Canada until the late 90s. And they got a foothold in a number of different areas, predominantly in the late 90s, but in the late kind of 2010s is when they really started to take off. And to understand why we might be interested in this particular crab on the West Coast, it's important to understand the ecosystems that it's invading. So you can see here a voracious, angry, aggressive green crab in a really nice seagrass meadow. And these seagrass meadows are made of sometimes one type of seagrass or sometimes multiple species of seagrass. They make up these really beautiful kind of grassy fields underwater, but they're flowering plants, one of the only flowering plants that lives completely submerged. And they have a full root system, just like other plants that you would see on the land do. And so they need a soft sediment in order to dig down and establish themselves. And unfortunately, European green crabs love soft sediment because they burrow into it and they make themselves these little kind of hobbit holes in the soft sediment. And in that process, they cut down or uproot all of the seagrass in the area. And this is a really big issue on the West Coast because seagrass is a massively important environment for lots of juvenile fishes as nursery habitat. Particularly juvenile salmon really use eelgrass meadows as a way to forage for food and protect themselves from predators before they get big enough to be able to kind of fend for themselves. And we have lots of organisms that rely on the seagrass either as a resident who has to live in the seagrass for protection or a visitor who will come in and out of the seagrass. But in general, the disappearance of seagrass has been coined as one of the greatest threats to salmon that you've never heard of. And so we're really worried about what the European green crabs are going to do as they're moving into these systems. And so in terms of what we know about trapping them and removing them from the environment, in the Atlantic populations, we actually know quite a bit. They have been there since the 1950s, and people have been worried about their impact on the ecosystem for quite a while now. And so we have a really good idea about herring, which is a nice kind of high energy content, oily fish, which the students from last summer can tell you does smell pretty bad. That herring is a recommended bait from our government, as if you want to trap European green crab, you should be using herring. And there is some debate about how efficient it is in the Atlantic populations or if it's the best possible bait, but it is still our recommended bait from the government. In the West Coast populations, where we've really had these issues with European green crabs establishing since the late 90s, but also into like kind of bigger problems in the late 2010s, your guess is as good as mine as to what we actually know about trapping these crabs efficiently, because we know that there are behavioral and genetic differences between our West Coast crabs and the East Coast crabs. But the West Coast crabs are so recent that we don't have a lot of published literature about their behavior, their history, what we can trap effectively with them. And so these students came out to the Banfield Marine Sciences Center. They started to explore the areas around the station, getting used to what kind of new environment they were in. And they started setting up their crabitat, which was a way to keep our crabs healthy and happy while we were waiting to run experiments on them. Because especially with behavioral experiments, if you have a happy animal, then you're going to get as close to normal behavior as possible. And so they started setting up all the crabitat and getting ready for our experiments to get started. And so we went through a workshop about field biology at the BMSC for the McMaster students. We talked about how we generate hypotheses. Basically, how do we turn an observation from what we see outside into something that can be a testable scientific question? We talked about things like experimental design and best practices for how we can conduct and design robust experiments. So we went through some workshopping. And then this is maybe not my favorite part, but one of my favorite parts of the whole experience is that I left all the students in a room with Sunita and a couple whiteboards and a few pens, and I said, design an experiment in groups. And I took photos of the completed boards, which you guys probably are upset that I'm including. But I did this because I came back after I had been working on a few other things. I came back into the room and they presented, each group presented their board. They told me what they wanted to do, what they knew, what they wanted to find out. And I was so impressed with what they came up with that I told Sunita, I said, I need to take photos of this because some of these are like master's thesis level ideas. And I want to see, we are going to be able to do some of them. And I'm really excited about it. But they detailed out what they were interested in and designed experiments around what they wanted to do. And in particular, it might be a little bit hard to see, but this setup right here on the bottom is actually what we ended up doing for one of their experiments. So they got to run 2 different experiments to look at the impacts of what baits were offered on the choices that green crab made. And the first experiment was dictated a little bit by what my summer students in my course at BMSC had been doing previously. So my students had talked about wanting to understand the differences between prey preferences with green crab when they're given 2 different choices. So herring, our recommended bait type, versus other bait types that are more readily available to the Hawaiian First Nations in their trapping program that are also, hopefully, fingers crossed, less expensive for them to use. And so there was one bait option that hadn't been looked at or used by my students in the marine sciences class, and it was the salmon head. And so the McMaster students were like, well, we can figure out a way to test if the salmon head is better or worse than our herring option. And what they ended up doing is making, I don't even know what we ended up calling it, like a head-eye slurry smoothie kind of concoction where they broke down constituent parts of the salmon heads and made a bait option for the crab to choose between the herring or this salmon eye thing. They were very brave, by the way. They dealt with a lot of things that smelled awful in the 2 weeks that they were in Banfield, and they did it all with a smile. This was the second experiment that they were able to come up with as not just something that they designed from scratch on those whiteboards, but also that was feasible and realistic in the timeline that we had. And what we ended up doing was addressing a couple of different things that the marine ecology students weren't able to get to. So the McMaster students wanted to see what the impact would be in a more natural situation where we had group dynamics of the crabs rather than just looking at one crab at a time, choosing between 2 food options. They gave them a longer expanse that they had to travel to get to their food. In their mesh divider, instead of just having one mesh divider that was removed that the crabs could then easily access the food, they kept the mesh divider in place and they cut a hole in the middle to simulate a trap opening so that the crabs, even if they got to this divider, still had to make the decision to enter through a door in order to get to the food. And then the big question they were answering with this was, does the age of the bait in the trap impact the crab's response to that food? When we set traps to get crabs out of the environment, we don't have the person power to be resetting them every 2 hours or every 6 hours. And so understanding how aging that bait in the environment impacts the attractant to the crabs is a really important element for how we can integrate that into a trapping program. And so the students got ready, along with the crabs in their spa day, to number all of the crabs and get used to handling them. These are really voracious crabs. They're very aggressive and lively, and they take a little bit of practice to get used to handling. And so the students were able to successfully plan a little spa day for the crabs to get all of their numbers painted on. And then once all the crabs were ready to go, we started prepping their baits. And so getting used to handling the crabs, but a couple in particular, I'd like to point out that a beer has an eye that she's pulled out of a salmon head here as they were prepping the eye smoothie. And Jonathan's got some fish heads, I believe, in his hands as they were prepping all of the bait options. So this very much was exactly, for whatever reason, what the students wanted to do. And they did every element of this experimental design. They had a question, they chose how they wanted to answer it, they got to prep every part of the experiment, regardless of how bad it smelled. And then also, while they were getting their experiment ready, they had the opportunity to talk to a number of the students that were in the marine sciences programs at the BMSC, where they were taking classes for credit. And they attended a science communication showcase where we had students that had done a whole bunch of different approaches to their projects that were related to their research. In particular here, this is a story time where some of my students wrote and illustrated a children's book about how green crabs came over from Europe and established themselves in Canada. And they also got to attend the Scholastic Symposium that these students were presenting all of their research at in like kind of a conference setting. And so it wasn't, I think having everybody out at the research station, again, wasn't just about going somewhere, doing an experiment and going home. It was really about interacting with and also meeting a lot of other scientists and researchers that were there. And so we got our experiment set up and running. They were so diligent with the way they set up everything from their experimental timelines, their schedules, how they wanted to run the experiments. Everything about this was driven solely by this cohort of students. And so they had their own lab space where they were set up with the crab holding outside, experimental space inside. And they ran through both of these experiments in the timeline, I think, from when you started to when you finished your experiments was like a week. Everybody was working really hard and really diligent and was able to get so much data in such a short amount of time. It culminated in what was known as our crab funeral. So as much as it would have been easier, to do your data collection and then go home, the students really were involved in every single step of this experiment, up to and including the permanent removal of these invasive crabs from our ecosystem through a humane euthanasia process. But what you can't tell from this photo is they were actually playing in the arms of an angel in the background as well. There was like a very solemn moment for the crabs. And so you might be wondering, after all of this, what did the students actually find? How did we help the Hawaiian with their trapping program? And where these results were actually really interesting to come across was that there wasn't a prey preference in these West Coast populations between the eye or head slurry and the herring, despite the government's recommendation being that we should be using herring to trap. One of the reasons behind this is that European green crabs are very voracious and they eat a lot and they are aggressive, and maybe they just eat the first thing that they come across. We saw no preference between herring and any of the other types of prey that we baited. And what this means for the Hawaiian First Nations in terms of a actionable step forward is that they can use whatever is readily available and less expensive to bait their traps with, which allows them a lot more flexibility in their trapping program. From the second experiment, the one that the McMaster students designed from the ground up about aging the bait, is that we found that there's actually no impact of aging bait for up to 24 hours. So the students were able to test fresh bait, 12 hours, and then also 24-hour aged bait. And we found that there was no difference in how the crabs reacted to these bait options, which also means that the Huayat can leave traps for up to 24 hours at this point, knowing that that bait aging, if they're using salmon head, is not inhibiting the attractants of European green crab. There will still be crabs that will be willing to move through a trap opening to access bait that's being aged up to 24 hours. And it was also, I think, again, really important to emphasize that it wasn't just about the science and the experiment that the students did. Even though they designed really amazing and robust experiments, did a bunch of data collection, they worked really hard, but they also had the opportunity to explore new areas. This is our group over top of a dogfish tank. So we had some dogfish researchers on site, and we were able to see some of these animals up close. We had, and I know that I don't have the pictures from this, but I know that the students were in the rowboats all the time, going across to other areas of Banfield, exploring the beaches. And as hard as they worked on this science, they also made sure to give themselves the time to explore these areas as well. And I think it's really special to kind of be able to access these environments from the base of a field station. And so where that leaves us is that we want to do this all again this summer because we had such a good time doing it. And through the Office of Undergraduate Research, we have the ability to bring a new cohort of students to a new field station this summer in the Kananaskis area of Alberta, which is in the foothills of the Rocky Mountains. And part of our experience with our field school this summer will be linked with those one-unit experiential learning courses that Pat mentioned at the beginning. This is the Kananaskis Biogeosciences Center. It's this nice set of buildings in kind of a foothills forest with a beautiful view of Barrier Lake, which is just across the road from the field station. It's a Biogeosciences Institute associated with the University of Calgary. And the access that we have to freshwater ecosystems, forests, alpine meadows is kind of unparalleled from this field station. In particular, this is a group of students that I had out there a few years back. This is the equivalent of like your science quad. It's right in the middle of the field station and kind of within all of the buildings. And we are able to access just a ton of different environments. Alberta used to be underwater as part of the inland sea. And so there's a ton of amazing fossil sites through Kananaskis. We have some areas that we'll go to see like marine coral and snail fossils that are really close to the field station, and also be able to access a lot of different forest and alpine meadow areas. And so these photos look a lot different from obviously where we were going at the Banfield Marine Sciences Center, but we're going to be focusing our project this year on a forest ecology question. So you can imagine that with the landscape that we have in Alberta, especially through the foothills and up into the Rocky Mountains, we have a number of different trees that are growing naturally here that are under threat to different pests and predators, and also climate change itself and the way that climate change accelerates pest invasion. And one of the major factors that is of interest in forest ecology is how can we estimate the drawdown of carbon from our environment and the storage of carbon in trees. And there's a relatively unique way of doing that, and it's very easy to estimate large-scale carbon storage in forest plots with very simple measurements. And so the group this summer will be coming out into Alberta, prairie grassland, and alpine environments in a carbon storage meets forest ecology style research project. And we'll be using these one-unit experiential courses as a base for you to also be able to get transcript record of the work that you've been doing. And we haven't solidified which ones they'll be yet, but we're thinking of something along the lines of 1 unit in experimental design and research methods, 1 unit in data collection, and then 1 unit in data analysis as well. So that's kind of the structure that you can expect the summer program to look like this year. And so I wanted to keep this somewhat short so that there'd be lots of time for questions. But I really did want to thank all of the students that worked on this science last summer because they really did an incredible job. And I'm so proud of the work that they did. And none of this would have been possible without Dr. Nadella Abir Siddiqui introducing me to this incredible community, as well as the Office of Undergraduate Research supporting our ability to continue to work on this. So I will leave it there. There's a little feedback form if you'd like to fill it out. You can scan that QR code with your phone. And then again, if you want to check out those 1-unit experiential courses, I've just got the link up here. But I wanted to leave as much time as possible to chat with you about the experiences and answer any questions that you might have. Thank you. Yeah, that's a great question. For those in the room who didn't hear it, the question was, how did we assess preference? Did the crabs have a consistent response, or were they always going for one fish over the other? And we had a large enough sample size across all of the bait types that we were testing where every crab was presented with herring or a salmon byproduct. And that was salmon carcass, salmon roe, the salmon eye slurry, and then we had another one which was a cheaper option than herring, and that was using chicken from the store. And we ended up having the crabs be presented with these options and recorded a lot of different behaviors around how they reacted to it. But our major one from an applied trapping perspective was, what do they go for first? Because if a crab is going to choose a prey, it does have to make the decision to move through an opening in a trap, which means that if we have one prey that attracts them to do that more often than others, then we could assign that as a preference. So in this case, the crabs had a whole bunch of different behaviors associated with what they were actually doing in these tanks. Some of them would run straight up to a piece of food as soon as it was like allowed to access it after the barrier was lifted, and they would grab that piece and they'd eat it. Some would go up and grab the food and then run to a side and then eat their food over like at a side of a tank where they felt more protected. Others would run up, grab one piece, and then double fist and run away. Like the behaviors that these crabs exhibited were actually really varied. And in particular, one of my favorite ones was when they were presented with roe, they would grow up and grab one little roe in each claw and then go to the side and like pop them like boba into their mouths. Like it was just, they had so many, you might not think of crabs as being like a personality hire in the ecosystem, but they had so many different like behaviors and exhibited such like unique characteristics. But the way that we actually assessed their preference in this case was, what did they choose first? So if they ran up and grabbed the salmon and then ran to the side, or ate the salmon and then ate the herring, or ate the herring and then went back for the roe, we would take their first choice as the one that they went for. In the thoughts that if that was herring or roe or salmon in the trap, then that would be the thing that would attract them most. Yeah, great question. Thank you. Yeah, so the physiology of European green crabs is actually pretty impressive. They're like these superhero tank crabs that are very robust. So they can survive out of the water for 5 days. They can survive in fresh water and in slightly salty water and in salt water. So they mostly came over through human-mediated transport of like shipping. The larvae are planktonic, so there's also the possibility that was brought up in like ballast water. But it was all, as far as we know, human-mediated dispersal. Yeah. So the data was first and foremost for the Hawaiian First Nations. They have given me permission to disseminate and talk about the results, so that's why I can be here today chatting about it. They had access to the data first in a technical report, but they are also supportive of us publishing it. So we don't have access, like, I don't have the skills to make a really cool website about this, but we don't have any of that available online. But there will be a publication that details all of the results, hopefully in the near future. And not that we went into this thinking that this would be a publishable study, but one of the really great side effects of having such dedicated researchers and the way that they design their experiments is that when I do get this publication up and running, everyone on this list is going to be an author on the paper because they were integral in the way that it was collected and designed. So I can't guarantee going into a project that will get publishable results from it. But that cohort did a great job and collected really good data. So we will get a publication out of it, hopefully in the near future. Oh, that is such a great question. So I have heard that green crab is actually quite tasty. The problem with so we have a couple different things here that you've touched on in this question because it's something that we think about a lot, and it's not predated upon by predators that exist on the West Coast in these natural ecosystems already because they are really aggressive. So they put up a fight before they'll get eaten, which can be quite damaging to anything trying to eat it. And then they also don't have as much meat on them as our native West Coast crabs do. So Dungeness crabs, red rock crabs, those are very tasty, very meaty, kind of slow, not the most aggressive crabs in the world. So when you're choosing between crabs, if that's part of your diet, you've got an aggressive, skinny crab, relatively, and you have a more docile, larger crab with more meat. And so predators do naturally go for the Dungeness or the red rock or a bunch of other of the crab species that we have on the West Coast. But I have heard that they are a tasty food option. And the problem with advertising it as such is that as soon as you commercialize something, there's the worry that people will try to start their own businesses in it, which would mean cultivating or intentionally introducing green crab into areas. And aquaculture is a really important element of food supply and production, particularly in Canada, but it's not without its flaws in terms of security of the system. So as soon as we set up if we ever had someone who was able to set up a green crab fishery, from an aquaculture perspective, you would have to just assume that they're going to get out at some point. So there's an element there that if we did have a productive money-making commercial element of green crabs, the worry is that it would steer into we should have more green crabs in our environments. And we're trying to avoid that. Yeah.
I have a question too. It's about the optics for the public. Do you know, I mean, I guess it's relatively new, their arrival in the West Coast. How do you know what the public feels about the green crab being killed off, basically, right?
Yeah, like what's the public perception of removing an organism from the environment with the express purpose of euthanizing it? You know what? That's a great question. I haven't done a lot of like community feedback work on it. I haven't read if there's been any surveys done on it. I believe that the messaging, particularly once the West Coast invasions hit, the messaging was already established that they were doing really bad things on the Atlantic coast. And so I haven't heard a lot of public kickback about green crabs are being killed and we're worried about it. There is, I think that DFO or Fisheries and Oceans Canada is really good about the way that they communicate on this issue. They have call lines that you can say, hey, I think I saw a green crab in this area. Can someone come out and look? They ask that you don't kill it yourself because it is possible to misidentify. And the last thing we want is people smashing a bunch of crabs in the intertidal because they look kind of like green crabs when they're actually some of our native species like a slender crab or a rock crab. But I think their messaging has been really clear about it. And I haven't heard a lot of public outcry about the removal of this species. What I have seen, though, is there's a lot of different groups that are very invested in removing it. So we got these crabs from the Clayoquot region, which is quite close to where Banfield is, from a company called Coastal Restoration Society. And they have three teams of people that go out and trap European green crab Monday to Friday. So every day of the week, they have 2 teams in the Clayoquot region trapping crabs. They have 1 team in Sook, which is further down the island. And in the Clayoquot region, because it's closest to Banfield, that's kind of where we can get our crabs in from. And when I was working with them back in October, just a few months ago, I was asking them, I was like, hey, like how many do you think we'd be able to get for our experiment? We're thinking, maybe 100, maybe 200. Like, what do you think that looks like? They're like, do you want 300? Do you want 500 males? We'll easily get that on Wednesday, so we can bring those over. I was like, no, I don't want that. That's too many. But they are trapping and pulling a lot, and that's every single day of the week across 3 different teams. They're pulling a lot of different crabs. So We've seen establishment, especially in the West Coast, in the Haida Gwaii region, which is north of Vancouver Island, and then also in other areas of Vancouver Island. We've seen really established kind of out of control populations. And so we also have now seen European green crab in the Banfield area. The Kuwait team has been trapping and pulling green crabs from that area, not nearly at the same rate so far, but we are still getting, maybe 100 a week in certain areas and then like 5 a week in other areas. So one thing I didn't mention here is that these results from the research that these students did last summer are informing a field study this summer with the Huayet, where they're actually going to be going out and testing this preference data in the natural environment. So they're going to be varying the baits that they set in their traps, and they're now using their program based on the results that we have to see how that translates into the actual environment that they're trapping in. It's a very long-winded answer, but that's a great question. Yeah, so it was a hybrid between those two populations. So one was founded based on a northward migration from the eastern seaboard, kind of in American waters, and it came up from those southern waters. And then the other population that came over from Europe later and established in Nova Scotia was from a more cold-tolerant population in Europe. And so those two subpopulations of the green crabs created this third, very tolerant population. I'm not entirely sure if we have the information, and this might be out there and I just don't know, but I'm not entirely sure which population, or if we know which population founded our West Coast invasive populations. It is absolutely possible, though, that through exposure to things like more frequent or intense heat events in the West Coast. Like we had a really bad heat wave that came through in 2021 that resulted in a lot of mass mortalities on the West Coast across the board. Exposure to things like that can heat harden organisms, where if they have been able to survive that heat event, then they might be able to survive subsequent heating events, and you can build up tolerance that way. If those heat tolerant traits have a genetic basis, and those can also be passed down to offspring potentially. So we do see the potential evolution of this trait on a relatively rapid time frame. From a crab's perspective, they live about 4 to 7 years, these European green crabs, and they produce hundreds of thousands of eggs per female per year. So their ability to pass on those tolerant genes and migrate southwards is absolutely a possibility. And we are seeing them, they started on the West Coast. We have a really well-established invasive population in Haida Gwaii. Now we're seeing more on the North Island of Vancouver Island, and now we have more introductions. There's been kind of 6 separate introductions that we've had records of in BC, and they kind of cover the coast from Haida Gwaii down to Vancouver Island. It's totally possible that as they get more tolerant, they'll start to migrate further south as well.
Okay, so please join me in thanking Dr. Smith again. And
Thank you.
I'm
Please, yeah.
Good.